guys, we're going over genetics, so go to page 30. I'm going to go over these short notes here. So these notes here just like act like genetics is like just two little fill-ins. Y'all, genetics was hard. Remember SpongeBob genetics? We did that for like 2 or 3 weeks. If we would go back to our review sheet, I actually think our review sheet has a lot of even better information. So here's what I have up here. To examine what traits will be passed on to the next generation, you need to make a Punnett square. We'll go over Punnett squares because, guys, you probably will need to make a Punnett square for almost all of these. Okay? Everyone has 2 copies for every gene. You get 1 copy from your mom, 1 copy from your dad. And remember, it's a 50-50 which one you get. Everything is random. Genetics is random, okay? This combination of alleles is your genotype. So your genes. What do your genes say? What you look like is your physical phenotype. So if my genotype is big B, my phenotype might be brown eyes. Okay, we'll talk about that. So dominant alleles are shown by using a capital letter. Example, if you have dominant alleles, you can be 2 dominant, or you can have just 1 dominant. But picture the dominant being, the darker allele. Like picture the lowercase being like white paint, and then the uppercase being like a dark paint, like red or blue. If you put a little bit of color in that white, can you ever get that white back? No. So if you have even 1 dominant allele, you are now dominant. You are no longer that recessive. 30. Recessive alleles, which are traditionally hidden, are shown by using, and it's 2 lowercase, such as little b, okay? Because there's no other combination that does not have a dominant allele in it, okay? Now, one thing I'm not including is when you have the same, you are considered homozygous. This could be big B, same. This could be little b, same. Okay? When you are different, so when they are different, it's considered heterozygous. And the only way to be different is a big and then a little, because they're different. Okay? Okay, anybody still copying? Okay. Now this is all coming back to y'all? Okay.
Somewhat.
Yeah.
I remember SpongeBob Susie.
Sponge Susie Round Pants.
Exactly.
Yeah. Okay. So, this is what's on our review sheet. Homo, same, hetero, different. Dominant needs a capital. Recessive are no capitals. So dominant can be capital, lowercase. As long as it has a capital, you read them together. Genotype, genes, phenotype, what you look like. Okay, so now we have basically 3 types, 3 combinations you can be. You could be big, little, or little. Okay, so remember, we have to call each of these either homo, hetero, dominant, or recessive. So is big, is that homo or hetero? Homo. Because it's same, think of like same, okay? And is this dominant or recessive? Dominant. And you read them together, good, dominant. So this is a homozygous dominant parent. What about this one? Homo or hetero? Hetero, because they're different. Dominant or recessive, you read them together. Dominant, good. And this homo or hetero? Homo, because they're the same.
What's there something about recessive?
And then dominant or recessive?
Recessive.
Recessive, because recessive means hidden, there are no dominant. Awesome. Okay. So now I'm just trying to get y'all to the point where if they would say there's a homozygous dominant male for blue eyes, you know what that, or brown eyes, you know what that means. Okay. So look up here. To get Punnett squares, just in case you forget, you have two parents. Big B, little b, and little b, little b. Okay? You make a window. You don't have to do this. I'm just reviewing. You make a window. One of the parents are going to have their two letters here. The other one are going to have their here. So I'm going to just pick another color and say, okay, big B, little b, they're going to be right here. Big B, little b. You don't put one here and one here. They stay on one side. Okay? Then I'm going to go to another one. Little b, they go together across the top. Okay? So bringing them down, you always want to do the big one first. Good. What's that going to be?
Big B, little b. Big B, little b. And then little b, little b. Little b.
Oops, sorry.
Fired.
So I see 50% are going to be heterozygous dominant, good, and whatever this phenotype is. 50% are going to be homozygous recessive, whatever phenotype that's going to be. Now, it is a 50-50 chance that your parent's going to give this allele or this allele, or this allele or this allele, okay? I do want to mention this. If this is one parent, does it matter which of these little b's you get? No. You know for certain that you're going to get a what? Little b, okay? Now say if you made it with this parent. Do you know which one you're going to get? A big B. It doesn't matter. So all the babies are going to be this. Big B, little b. Did you have to do a Punnett square for that? No. Sometimes you can predict, okay? Now, if you have this, big B, little b times big B, little b, you don't know. Because technically, you could get a big B, big B. You could get a big B, little b. You could get a little b, big B. You could get a little b, little b. There's a lot of different combinations. That's why it's always good to do a Punnett square, okay? So, if we had to do this Punnett square, this is the most diverse Punnett square. Big B, little b, big B, little b. Ooh. Dropping it down. Okay, you have a 75 dominant, 25 recessive. Or if we're looking at genotypes, you have a 25, 50, 25. Okay, let's do some practice. Okay. These are ones that you cannot do Punnett squares to. You have to use your critical thinking about what has to be given. So, for example, which trait will most likely be observed in all offspring of the above parents? Now remember, the only way you know something will get inherited is if it is homozygous. Because if all you have is a little or a big, you know that's all. So for example, this right here, you see how it's a big B, big G? Do we know what's going to be passed on?
The green feathers.
A big G. Now, a big L, a little L, do we know which one's going to be passed on?
The big L.
You know that? Does dominant always get passed on?
Not always, but it can.
Okay, so if you have this as a parent, we don't know if this one or this one's going to get passed on, but since they're both the same, we just know it's going to be a big G, okay? If it's a big L and a little L, Do we know for 100% certainty which one we're going to get? No. Okay, so do we know which G we're going to get? Yes. Do we know which L we're going to get? No. Do we know which G we're going to get? No. Do we know which L we're going to get? Okay, now, so we know we're going to get a big G. We know we're going to get a little L. Now, little L means short beak. Do we know that it's definitely going to be short beak? What if this parent gives the big L and now we're long beak? So do we know that the phenotype is going to be recessive if a dominant is over here and can be given? No. The only way we know something is going to be given is if you have two dominants and you're definitely going to give a dominant. So because we know this dominant allele is going to be given, we know it's going to have green feathers. Okay, do that one over there. I want you to actually right here tell me which one you predict. If you can't predict it, write X. Okay, same with this one over here.
So I wonder if we can read like 50-50 chance.
Nope, but that's not 100%. You can't predict a 50-50. So do the same thing for the next one, where we're using the parents' two genes. If they're homo, you can predict. If they're hetero, you can't. Good. Definitely predict.
I'm just going to put 50-50 where it's homo.
Okay, look up here. Because it's a big G and a little g, it's a 50-50, so there's an X. I don't know which one it's going to give. Big B, little b, I don't know. It's a half and half. But since there's two little g's, I know it's going to get a little g. And because there's two big B's, I know it's going to get a big B. Now, because it has a little g, I still don't know if it's going to get a big G from over here. So even though I know it's going to get a little g, I still don't know what kind of G is going to come over here. Now, because it gets a big B, I don't care if it gets another big B because I know it's going to have the big B dominant phenotype, which is green eyes. Okay.
So it's suffice. If you have like a big B and a little b, and then somewhere down the line where you have double B's, I wonder if that little B could still come but it wouldn't be dominant anymore.
Well, you can still have it hidden, and then if you would be with a recessive person, that could come out. Yeah. Okay, so there's a couple questions in here where if you understand what we're doing here, you know that they're talking about a homozygous dominant, you could predict the phenotype. Okay, let's see what's next. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 questions y'all got. Okay, you got 10 questions. I'm going to give you about 10, 15 minutes.
Do you want us to start on the two that we just did?
Yeah.
Or under the two that we just
Yeah. So like, hey, real quickly, skip to this one right here in the Cocker Spaniel. I'm sorry I don't give you a lot of room for Punnett squares. If you need scratch paper, you can come up and get some. But for example, it says, in Cocker Spaniels, the allele for black coat, big B, so black is big B, is dominant over brown, which is little b. So little b is brown. Okay. If a brown Cocker Spaniel is crossed with a hetero black, what are the genes for a brown?
Little b, little b.
Yeah, because they just said brown is dominant over. So a brown is little b, and what is a hetero black?
Big B, little b.
Yeah, so now they're not giving you the B, so you need to figure out it yourself. Okay, so I'm going to give you 15 minutes, then we're going to go over these.